Hello, everyone, and welcome to today's webinar, Shifting Tides in Dementia Research, Targets, Technologies, and the Changing Landscape of Pharmaceutical R&D. My name is Colleen Lezovitz, and I am a Senior Marketing Manager at Worldwide. Before we kick things off, I wanted to go over a few things. All attendees are on mute, but please use the questions window to submit questions at any time pertaining to the topics being discussed, or if you experience any technical difficulties. I also wanted to point out that we have a couple handouts available for you to view, including an Alzheimer's ebook and a Pharma Voice article, and you're able to access those in the handout section of your console. All attendees will receive a link to access the on-demand version of this webinar, so keep an eye out for that, most likely tomorrow. I would like to go ahead and now introduce our experts. You'll be hearing from Michael Murphy, our Chief Medical and Scientific Officer, Natalia Drosopolo, who is a Senior Director of Project Management Neuroscience, Hank Reardon, who's Executive Vice President of Scientific Solutions, and Tom Babich, who is our Vice President International of Neuroscience. Before handing things over to Dr. Murphy, we would like to hear from you, our audience, so I'm going to go ahead and push out our first poll question.
Well, thank you, Colleen. So, ladies and gentlemen, welcome to an intriguing topic. And what we would like to do first, as Colleen has emphasized, is get a bit of a better understanding from your point of view, your research interests. So here it is. Do you have a current research and development effort which focuses on neurodegenerative disorders outside of Alzheimer's disease? And the choices are: you do not have an R&D effort in neurodegenerative disorders at all. We have on these webinars many times people who are just interested in the topic but have no active technology in that target. So that's one choice. A second one would be, we do have ongoing R&D, but it is confined only to Alzheimer's disease and related phenotypes. Lots of activity in the industry currently in that domain. And then finally, an interesting one is, yes, we have ongoing R&D, but it isn't limited to Alzheimer's disease and related phenotypes. So we're asking you to please vote for one. Colleen?
Okay, I'm going to give everybody a few more seconds to submit your response, and then I'll go ahead and close it and share the response.
Interesting. Well, you know, I think ladies and gentlemen, I think we anticipated the audience beautifully, because we also feel there is a shifting tide in research toward other neurodegenerative conditions, not limited to Alzheimer's, and it's encouraging to see that constitutes a very nice percentage of our audience. We begin with the next slide, which I hope captures the sentiment of where we think we've been and where we might be headed. Now, this picture really talks about the optimism and the enthusiasm that those of us who've been in this venue now for over 40 years have brought to the field, both in discovery and development, kind of living the life, innumerable compounds, many different hypotheses and compounds, many different studies, contributing with a large number of other individuals to what I call the science and the art of clinical research. And today's presentation, however, is not about where we've been, although we could regale you quite a bit with a lot of neat anecdotes, I think. It's more about where we're headed and what does the future hold. Is it one of promise and opportunity? How do we see it? Next slide, Colleen. So for those not familiar with Alzheimer's, just to give you a sense of the area, this is a real public health care problem. So there's 75 million people estimated to have a dementing syndrome by 2030. If you look out to 2050, the numbers are staggering. So the magnitude of the public health problem cannot be overstated. Aging population, huge impact on patients, families, healthcare systems. It's always provided a call to action. A call to action. Many etiologies, as we know, under the big umbrella of dementia, there are a number of different indications, many different etiologies. Increasingly innovative technology. I'm sure you'll agree with me, in the last few years, the technology has never been more exciting. I could not have imagined this 15 years ago. Just an amazing portfolio of ideas that people have generated. Diverse presentations. Tom will talk about that today. for a family of illnesses, increasingly complex trial designs, particularly for early phase investigations. We'll talk a bit about it, that help us understand the mechanism of action, the dose, the probable clinical targets. Really interesting. And importantly, on that right-hand column there, very complex societal, personal, and economic impacts, a mosaic of mediating and moderating variables that impact the entire society, genetic, environmental, lifestyle, and so forth. All of those are moderating influences on maybe the genesis of these disorders, not to mention how they present. The next slide kind of illustrates the spirit of this. I first encountered this back in 2009, and I have to say it reminded me of the old axiom that many of us use, a bit of humility, reinforces the notion that, the more we seem to know, the more that we don't. This remarkable slide is from brains of two different monozygotic twins. Monozygotic twins. You're looking at staining here for amyloid and tau, who was nevertheless discordant for Alzheimer's disease. Both raised in the same family, both in the same community, presumptively similar diets. Interestingly, both went to the same university, both became engineers, both became chemical engineers. But the gentleman on the right, he developed Alzheimer's disease at age 60 and he died at age 76. The gentleman on the left, also a chemical engineer, identical environment, education, and so forth, died at age 79 from prostate cancer. So what does this say? Is there an epigenetic influence across all the different hypotheses that people usually tout for Alzheimer's and related disorders? Is that a moderating influence? It certainly suggests to me that there are gaps in disease mechanisms and risk. This is part of the NIA initiative, of course, to fill in those gaps, to inform the basic pathophysiology so that we can have druggable targets. It confirms what we all acknowledge, a need for better patient segmentation in terms of who we enroll in studies or exclude. Does this mean that we need targeted therapy with smaller preclinical black boxes? Now, the literature would actually argue that phenotypic drug discovery efforts have been more productive in CNS, not targeted approaches, but nevertheless an interesting debate here. Does it argue for smaller preclinical black boxes? Or maybe it brings up the whole notion that we've all heard about of cocktails and lifestyle modifications. The bottom line I want to get to is that little expression on the bottom. I think this slide reminds us, as we move beyond Alzheimer's, that we need to build the bridges more slowly in order to cross them more quickly. Building a bridge more slowly to cross them more quickly. Now, in the next slide, we illustrate all the different resources that we have to help us do this. Terrific support systems in government and academia. All are familiar with the NIH research portfolio. They have been so enabling for many of the initiatives which pharma has pursued. The patient advocacy groups always been a supporting force, increasingly, however, used to help us with designs and the assessments. Those of you that work in the orphan space will recognize that immediately. We want the families and the advocacy groups to assist in helping to design these studies in a way that equally informative, that are more conducive to a patient and family's lifestyle. Many of you may be involved, for example, with at-home assessments. We call them site-less clinical trials, an interesting initiative. And CROs, what about those folks? Now, the job of a CRO, in a way, is to offer predictability and efficiency to what I would call transition to an inflection point. An inflection point for us is a point in clinical development at which a decision is enabled. By approaching that point with rigor, with efficiency, we permit more compounds and more companies, frankly, to have an opportunity to evaluate remarkably different mechanisms of action. And we've learned a great deal. Lessons learned have been astounding. The opportunity to see what we've done in the past versus what we'd like to do in the future is really what this presentation is about. And on the next slide, you'll get a sense of the three different topics. We had lots to choose from, but these are the three that we thought were most informative. There's the section on patients. Tom will deal with this. What are we doing in terms of patients by disease state, by duration of illness, by presentation? We're all familiar with this. Are we orphanizing the dementia space? That's an interesting topic. Orphanizing dementia space. Hank will bring in the biomarker thematic, both pharmacodynamic and disease-related measures, which are particularly important in the early stages of evaluation for mechanism of action and utility in patients who otherwise do not have clinical signs of these illnesses. The pre-symptomatic individual who does not yet demonstrate clinical signs or symptoms, but may in fact have a long-established biological pathophysiology in place. And finally, Natalie will talk about operations by illustrating a recently completed multi-center study in an unusual indication that emphasizes for me the fact that differentiation is in the art of trial conduct, the ability to encapsulate a research question into a standards of care. I really like this notion, encapsulating a research question into a standard of care, which, as we know, can be radically different across the international environment where all of us increasingly work. So having said that, Tom, let's look ahead, if we could, looking at optimizing clinical trials in dementia with Lewy bodies and Parkinson's disease dementia.
Good morning, everyone. I'm going to spend a few minutes trying to share with you my basically my doubts and dilemmas regarding neurodegenerative disorders, and also I will address similarities and differences among several most important and most frequent neurodegenerative disorders, where we basically what we have in our everyday practice. In the next slide, you can see that practically all of these neurodegenerative disorders are related to three different proteins, to accumulation of three different proteins in various parts of our brains. On the left-hand side, you can see that alpha-synuclein can be pathologically accumulated in subcortical area and brainstem, whereas another molecule, which is a tau, can be making some specific neurofibrillary tangles, can be in nerve cells and mainly accumulated in the cerebral cortex. In all of these conditions, we can find a substantial number of amyloid deposition, which is a main feature of Alzheimer's disease, but also amyloid can be seen in healthy people as well, and as well in people with predominantly synucleinopathies. So the knowledge of neurodegenerative disorders are progressing fastly, and when I was a student 40 years ago, Alzheimer's disease was orphan disease. And also regarding all of these synucleinopathies and tauopathies, with the exception of Parkinson's disease, practically all of these disorders are becoming very popular in the recent 10 years, where our knowledge about pathology, pathophysiology of these disorders is going to rise. However, all of these conditions are very complex and presenting with several different syndromes at the same time, and several subtypes and phenotypes. So for instance, in case of progressive supranuclear palsy, which is the typical tauopathy with a Parkinsonian expression, there are so far 7 different phenotypes. In a frontotemporal degeneration, similarly, there are 6 or 7 different phenotypes as well. In dementia with Lewy body, for instance, there are 7 different syndromes which are basically creating the disease of dementia of Lewy body. So in spite of the rising knowledge, we are still in some gaps how to make all of this knowledge together and how to create rational clinical trial, how to create a rational drug development program because of the complexity of all of these disorders, various phenotypes, and lack of all comprehensive knowledge about neurobiology of these conditions. We still have a problem to confirm in confirmatory clinical trials the efficacy of the molecule which we believe that might be helpful in our patients. It's important to say here that basically all of these conditions are presenting with two basic syndromes. One is a cognitive impairment, and another one, it's a Parkinsonism, or if you want to be more precise, bradykinesia and rigidity. And in fact, many of patients with Alzheimer's disease will finish their life in a condition resembling to Parkinson's disease, whereas many of Parkinson's disease patients will develop a classical picture of Alzheimer's disease at the end of the course of disease. So that's why there are a lot of overlappings among all of these conditions in terms of pathology, in terms of clinical presentation, and because of the lack of biomarkers which should be specific for each of these disorders, we still have a problem how to make a diagnosis, particularly how to make unique, harmonized diagnosis for patients in clinical trials. So some of you who basically participated in various clinical trials before know that in the big clinical trials like bapineuzumab or similar, we had at least 30% of patients where we basically thought that there was no Alzheimer's disease, although the main indication was Alzheimer's disease. So how basically to cope with these problems and what we can do in the future to improve our knowledge about clinical trials, I will show you in the next slide. So for instance, if you see prevalence of various disease and epidemiology, we still have a problem how to understand what is the prevalence of such conditions. So for instance, we know that multisystem atrophy, progressive supranuclear palsy, or corticobasal degeneration has a low prevalence at the moment. But for instance, the most second dementia, the most frequent dementia after Alzheimer's disease, which is dementia of Lewy body, in fact, we have no absolute data of prevalence of these disorders. There are some impression that this be 25% of all dementia belonging to dementia of Lewy body, but recently published documents showing something different. This figure is lower than 5%. So anyway, we need real observational clinical trials to show us what is the real prevalence of some of these disorders, including frontotemporal dementia, where we have prevalence data from 1 patient to 33 patients in population of 100,000 people. On the other side, clinical circumstances of all of these disorders are still mystery for all of us because intra-disease variability, inter-disease overlapping, of course, and what is the most important, lack of specific symptoms in an early stage of disease. So all of these disorders are basically characterized with a long latency before diagnosis and a pretty short life of our patients. Very rarely our patients, of course, with the exemption of Parkinson's disease, can live with these disorders more than 10 years. So basically, we have no enough time, maybe 3 to 4 years, where patients are still in a condition where we can make a clinical trial, where we can find something among new drugs. So basically, diagnosis should be faster, better, with more accuracy. In Alzheimer's disease now, we are trying to basically include patients with no dementia in clinical trials, which we are calling shift to the left. So basically having patients who might have Alzheimer's disease in the future, but we still have a problem with the diagnosis of patients with Alzheimer's disease. It still takes a long time in all of sites. On the other side, any any diagnostic criteria that we are using for any of these particular disorders can overlap with something else. So if we use, for instance, criteria for diagnosis of Alzheimer's disease, we will hardly rule out all of patients with frontotemporal degeneration. On the other side, if we are using criteria for progressive supranuclear palsy, we will definitely have inside some patients with multisystem atrophy who also has a certain percentage of dementia, which was really new knowledge recently published that practically patients with multisystem atrophy when have a between 15 and 30% of dementia syndromes inside. So anyway, we are in a position that we need desperately to improve our diagnostic tools to provide more sensitive and more specific diagnostic tools using biomarkers and particularly neuroimaging. At the moment, we cannot be satisfied with the biomarkers in the CSF and blood and also in neuroimaging. That's why we need more randomized controlled clinical trials. We need more data to have a consensus about standard care. And also, what is very important for all of these conditions, we need a sensitive global outcome measures, because at the moment, if you see in progressive supranuclear palsy, in multisystem atrophy, or in Parkinson's disease, we are using quite a global outcome measures, whereas in some conditions like dementia Lewy body, we have no comprehensive measure to assess the effect of our product on disease on disease modification. So that's why we are still in a position to take a great challenge in the next few years trying to improve our skills, tools, and knowledge regarding neurodegenerative disorders. I would stop here if you have some questions, or if we can share with you some of our questions.
Oh, we do, Tom, and I'm going to start out with one because I had a Yogi Berra moment here, kind of a deja vu all over again. So I want to see how you think about it. I think there are lots of uncertainties here, targets and methods. It seems like just a labyrinth of issues that need to be sorted out. So I have kind of two minds of it. I wanted to get your opinion. So on one side, do you think this argues for a more cautious, conservative approach to clinical development, more exploratory research rather than confirmatory studies? Or on the other hand, does the business need of the industry, not to mention the unmet clinical need, prompt us and propel people to continue to kind of skip over, so to speak, phase 2 and get right into phase 3? I remind you, Tom, back in the day when we were doing the cholinesterase inhibitors 30 years ago, we had the same issues. We would argue about everything, how to make the diagnosis, what are the assessments, and so forth. But it didn't prevent the industry from moving forward very aggressively. So what's your opinion, Tom? Do we go slow or do we go bold here on these programs?
Well, thank you very much for this question. I would probably remind you on when you mentioned the history of acetylcholinesterase inhibitors, I remind you on the history of levodopa in Parkinson's disease. When levodopa was developed 55 years ago and started in clinical treatment, we basically used levodopa as an antibiotic, which means the higher dose, the shorter time of treatment. And we realized, in fact, this is not the right approach, but because every of our patients developed side effects within 1 year. Now it takes 5 to 6 years to have this side effect. So basically, we understood that we need to go differently to treat patients with Parkinson's disease, which was start low, go slow, but go. And similarly, in clinical research, I think in a condition of neurodegenerative disorders, I think our clinical knowledge is still not sufficiently confident to be ready for confirmatory clinical trials, particularly in tauopathies, in alpha-synucleinopathies, of course, with the exemption of Parkinson's disease. So that's why I would rather collect more preclinical data, have a right translational way to the clinical trial, and start probably clinical trials and development from early stage with a very good proof of concept trial towards either tauopathy or synopathy. It's very difficult to find the right model. For instance, for tauopathy, many of companies now wants to go to PSP because this is the right model of tauopathy. Although I still believe that pugilistic dementia might be even better for that. But definitely, I think this is the right way. At the moment, I'm afraid we are not ready for confirmatory clinical trials in the neurodegenerative disorders of tauopathies and synopathies, a part of the Parkinson.
Hank, I know you have a point of view in this as well.
Yeah, Tom, just as a follow-up question to your last answer, you cited all the clinical complexities, including the kind of intra-disease variability and the inter-disease overlap. Given all that and what you just said, would you recommend when we finally do get to the point where we do registration trials, would you recommend more of a disease modification approach or just simply go after a symptomatic approach for drug developers?
This is a very good question. You know, it's very difficult to answer. This basically depends from indication to indication, because if, for instance, in dementia of Lewy body, if we target dementia, we will probably improve dementia in some drugs like cholinesterase inhibitors, but on the other side, we will probably make worse in neuropsychiatry symptoms or autonomic dysfunction. So it is quite difficult. At the moment, I feel that we have no enough molecule which will satisfy criteria for disease-modifying treatment in neurodegenerative disorders. And there are some molecules, anecdotally, which might be useful in some symptomatic approach of these patients, particularly treatment of falls, treatment of dyskinesia, for instance, treatment of autonomic failure. And this definitely depends on a development program of the companies, you know. But I think at the moment there is a more chance to have a success and a licensed product with a symptomatic approach than with the disease modifying. In fact, these clinical trials are shorter with more chance to provide data with proven efficacy and acceptable level of toxicity and patient safety.
This is a great transition to the next poll question, Colleen, if you could, please. Great segue. So within your R&D program, would you say your efforts in neurodegenerative disorders are strongly translational with a fully integrated preclinical clinical team, or would you say that your efforts are more traditional, kind of the traditional development pathway that we're all familiar with? So we're asking you to choose one here to give us a feeling for your corporate philosophy, if you don't mind. Right ahead, please select one. Oh, fantastic. Well, not a surprise given all of the interest in the indications outside of AD. Fantastic. Our sentiment working with many different companies that this is universally true, virtually universally true, particularly for the non-AD type of indications. This leads us into the next topic, which of course is of the biology of the illness and the emerging importance of biomarkers. So Hank, if you could, please.
Yeah, sure. Thanks, Mike. Well, even to casual observers, the role of various biomarkers has become increasingly important about assessing the relative safety, tolerability, and efficacy across a range of all these disorders, but especially Alzheimer's. And in drug development, you noticed, even as looking in just real quickly, we've done a lot more work in biomarkers for inclusion, outcome measures than we have done even a few years ago. It's very rare for us to do any studies at all that don't really have 3 to 4 different biomarkers at play at the same time. In some instances, these are predictive or prognostic or even trying to be surrogate biomarkers, but there's usually a biomarker in there. So although Tom is correct in that the specificity for these biomarkers is not so great, at least in AD, there's no shortage of biomarkers. In fact, most people we talk to about designing trials tell me they're biomarker agnostic, and that's their way of saying we throw everything at it and see what sticks. So unfortunately, because of that type of approach, and which is not a good link to a mechanism of action, the NIA and Alzheimer's Association recently adopted research criterion that helped utilize a classification scheme to help us with the most important biomarkers. So this was rolled out a year ago by Cliff Jack and group, and actually updated again in July of this year, and actually presented at AAIC, if any of you were there. And it was met, unfortunately, with a lot of pushback, which was a little surprising. And as we go through the system, you'll see why. We've also published a paper on this, and actually just came out yesterday. We're happy to present that to you. Because the goals of this kind of approach from NIA, AA, are to facilitate a more precise approach to intervention trials in which the pathways can be targeted at more precise points in the disease process, and importantly, targeted to the most appropriate patients likely to benefit. So very lofty goals for this, and was really meant to be a research framework used in observational and randomized trials, and not really viewed as general clinical diagnostic criteria. And I think that confused some people. This is what commonly referred to as the ATN system, in which we have 7 different biomarkers divided into 3 binary categories. In this scheme, the letter A refers to beta amyloid biomarkers reflecting binding of amyloid PET scans, or low CSF amyloid like A beta 42, which designates the presence of beta amyloid plaque and associated pathophysiology. The letter T refers to tau biomarkers, such as elevated CSF phospho-tau or tau PET binding, and reflects aggregated pathologic tau. And N represents biomarkers of neurodegeneration or neuronal injury, such as hypometabolism 18F, FDG PET, or atrophy on MRI, or even CSF total tau. Of note, there's both an imaging and CSF biomarker in each of the 3 categories, making it possible to have a pretty complete characterization of these biomarkers just using one modality, such as imaging or CSF alone. As a potential subject trying to get in a trial, you can either be positive or negative on each of these biomarkers, A, T, or N, and therefore there are 8 possible biomarker profile combinations, each corresponding to a label representing a range from kind of normal biomarkers to AD pathophysiology, to Alzheimer's disease itself, or non-Alzheimer's disease pathophysiology. So using this research framework, a subject who has a biomarker evidence of A beta pathophysiology alone, which could be either by PET or low CSF A beta or 40-42 ratio or whatever, but with normal tau would be assigned the disease label Alzheimer's pathophysiology, while the term Alzheimer's disease will be utilized only if there was biomarker evidence of both A beta and pathologic tau. Of note, in this model, Alzheimer's pathophysiology and Alzheimer's disease are not regarded as separate entities, but rather earlier and later phases along a pathophysiologic continuum. And importantly, once again, these labels have no regard to clinical symptomatology. So therefore, AD in this model could be viewed very exclusively as a proteinopathy in this research framework. So it really hinges upon the understanding and not acknowledgement that AD is an aggregate of pathophysiologic processes, and that could be defined in vivo by biomarkers, but not by clinical symptomatology. The separation of pathophysiology and clinical state is a real departure from the original 84 NIA criteria and even the update. So it's a shift away from AD first as a clinical pathologic construct, and then as a clinical biomarker construct, where the biomarker was used just as supportive. And that's kind of the key feature moving away. So it really is buttressed by our increasing awareness that dementia is not a disease, but rather a syndrome composed of signs and symptoms that could be caused by multiple diseases, one of them of which is Alzheimer's. Next slide, please. Well, the adoption of these criteria will have real clinical trial implementation issues. Of course, there's the expense of, PET scanning, and then the relative limited availability of scanners throughout the world, and big regional differences in that. And then, of course, even doing lumbar punctures, things that should be pretty easy to do, that application of LP technology and the right methodology varies regionally, even within countries, and really hinders us doing a lot of multinational trials. So the adoption of this framework obviously increases the overall price of doing AD trials, where in many of our studies, imaging and CSF alone now account for about 1/3 of the total budget, and importantly, increases the rate of screen failure rates, and basically for people not meeting the various thresholds for these biomarkers, even when they're combination ones, or you could do one or the other. So it's not unusual for us to see screen fail rates as high as 75, 85% in these studies which utilize some of these criteria, both imaging and CSF biomarkers, depending on what that criteria may be. So that means that, you know, we're screening many, many more people to get into trials. So we've developed a kind of hierarchical approach on eligibility factors that we conceptually feel helps us to really divide screening factors into 3 areas. Those are completely predictable, you know, things like age, medical history, progression of cognitive decline for a good site, and medical status. Those that are semi-predictable, such as cognitive test scores, the presence of depression, caregiver burden, those kind of things. And those that are not predictable, such as the biomarkers, you know, even EKG findings, but amyloid or tau and CSF or PET imaging. Unfortunately, the treating clinicians who are very familiar with their patients and caregivers, they can increase their knowledge of screen failures, making some of the semi-predictable ones more predictable and decrease their screen failure rates, whereas those sites that rely on advertising for subject recruitment alone may be at a relative disadvantage. So our job is to kind of move the semi-predictable to the predictable ones. It's of course much more difficult to reduce screen failure rates by non-predictable factors such as the CSF or beta amyloid or even safety brain MRIs like for ARIA. However, there are a couple new promising techniques that are out there that help render these factors more predictable by using different statistical tools that help predict the presence of tau or even the conversion of MCI folks to AD. These techniques vary widely. Some use adaptive designs, but they all center around really multiple regression analysis in the end to predict the presence or absence of beta amyloid or tau on imaging or CSF based on earlier scores obtained from less expensive and easier to get screening measures such as demographics, cognitive test scores, genetic status, even clinical signs and symptoms, and even MRI and CT findings are actually cheaper and easier to get than others. So we've been really trying our best to really help sponsors out by reducing that screen failure rate by using some of these methodologies. Any questions about that?
Well, I've got one, Hank. So, you know, I've read that ATN paper, I must admit, more than 2 or 3 times. There's also a companion article coming out that tries to interdigitate the biomarker profile with the neurocognitive profile. I can see it. But here's a question that's been voiced by a few. I want to see what you think about it. So the proposed guidance that we've just reviewed for biomarkers pivot strongly around A-beta and tau hypotheses as potentially causative, not simply associated with Alzheimer's, but pathophysiologically related to its genesis. Certainly, you can't get the diagnosis of Alzheimer's, I don't think, unless you're amyloid positive. Now, but doesn't the failure of the anti-amyloid products, hitting production, metabolism, and clearance, I've heard it said that, doesn't that introduce at least a note of caution in all of this? So do you think, for example, that the system is basically incomplete?
Yeah, so that's a good question, Mike. And in fact, the authors acknowledge the system's incomplete, and they build in as much flexibility into the system as they can by allowing the addition of other biomarkers. So like Tom was saying, other biomarkers that become increasingly important, like alpha-synuclein, are not really addressed here. And to be fair, some of that is because some of the knowledge has become just more recently available. There are no good imaging techniques, and they're really just really new to the framework, and so the reliability is uncertain. It's widely accepted that CSF beta amyloid levels and tau PET are valid indicators of abnormal pathophysiological state associated with tau deposition, but other biomarkers, for other proteinopathies like alpha-synuclein or even TDP-43 that play a crucial role have been largely overlooked. And in fact, you could really think that maybe the most important biomarker, which many neuropathologists think is very important and relevant in neurodegeneration, is microhemorrhages. But unfortunately, neuroimaging at this point is too crude to serve as a biomarker for this. And other things like neurofilament light change or things that measure synaptic degeneration, loss, or accidental injury, they're not specific either. So really it is incomplete, and by adding more biomarkers to the model, it really makes the model much more cumbersome, because then you have a lot more, forget the clinical staging goes along with it that I didn't mention too much, but also increases the possibility of having incomplete biomarker profile or discordant biomarker profile, which one biomarker, is in the opposite direction than the other ones that measure approximately the same type of underlying pathophysiology.
Natalia, I just heard your work in clinical operation getting a lot more complicated. How do you see this?
For sure. Thank you, Dr. Murphy. So yes, Hanka, for me, from an operational point of view, I'm more interested in how this new research criteria updated, what impact they have on the clinical practice, how much more difficult it makes our clinical environment.
Well, speaking of which, Natalia, I know you've had a recent success story in a difficult orphan indication and would like to hear your perspectives on executing it. If you could,
yes,
please.
Mm-hmm. Yes, so thank you. Well, I'm happy to say that from this successful completion of a recent complex and global program on frontotemporal dementia, which, as many of you may know, is considered a rare type of dementia, we have gained really valuable insight on how one can apply, let's say, operational efficiencies that can help us optimize the execution of other studies, especially in dementia. To be more specific, on the next slide, I can give you some examples on our operational takeaways from our recent experience. So firstly, with regards to organization, staffing, approach, we can really borrow experience from other disciplines and even therapeutic areas, I would say, such as orphan diseases studies, when one should have a more translational mindset, for example, on bioanalytics, refined phenotypes, even specialized measures. Another lessons learned from rare diseases studies one can borrow and apply on other studies, even dementia studies, is that we may need to be more innovative and adapt an approach to think outside the box. A good example is on a recent experience on patient recruitment. Conducting a rare disease study, one can imagine that patients are very difficult to find, and can several times mean that we may need to bring the patients to the specialized centers. A recent experience, we have managed successfully an early phase study where we had literally to fly patients with their caregiver and sometimes even medical support from different countries in Europe to UK, and similarly from different states in US to specific centers in order for them to perform PET scans, as the specific radioligand used for the PET scan was only available to be made for those few PET centers around the world. Similarly, I would say, utilizing our experience from different therapeutic areas, such as large cardiovascular outcome trials, we applied lessons learned on management and coordination of multiple moving parts to run a successful study. Secondly, I mean, very importantly, an operational takeaway is that the medical monitor engagement with site is key. It allows for the medical monitors to really possess a high level of indication-specific competency and resolve indication-specific issues in a more fast and efficient way. Creating a chief medical monitor, like a CMO, within the CRO for the study was found to be very beneficial. And finally, high effective teams are mandatory. Team members that are assigned in the study who are familiar with local languages, customs, standard of care, as well, and very importantly, the local national health system, more importantly in international time frame, within each participating country, provide a more effective overall study management and really contributes enormously to the successful execution of the study. And as in worldwide clinical trials, we're truly a global company and scientifically focused. As you know, we have the advantage to really offer our studies those highly effective teams. And that's, I don't know if anyone has any questions.
Well, you know I have one, Natalia. I don't think I've ever heard anyone use the phrase orphan disease and large-scale cardiovascular outcome trials in the same sentence. So what is similar and what is different about, large-scale cardiovascular outcomes trials, which of course we do quite a bit of, but so what's the analogy like to this particular indication as an example?
Well, Dr. Murphy, they are different in many ways indeed, but then again very similar in other ways. Similarities are more often in terms of operational approaches as well as management coordination. I'll give an example. Both orphan disease and cardiovascular outcome trials are usually global. They have to be conducted in large geographical region, but of course number of participating sites and enrolled patients differ. So in our recent frontotemporal dementia study I mentioned earlier, we had to go to about 12 countries, 65 sites to really enroll 200 patients. In the recent cardiovascular outcome trials we conducted in worldwide, we had 32 countries, 1,000 sites, and more than 26,000 patients. So scale is bigger in cardiovascular outcome trials, and this is very different from orphan disease studies, but again need the same approach of outreaching to multiple regions. I'll tell you another very similar feature. There are the multiple involved stakeholders. Apart from the sponsor and the CRO, you often have to partner with academic research organization and patients advocacy groups, as you mentioned before. All the stakeholders, of course, have the common goal of the study to succeed, as we all expect, but may have conflicting agendas, which may raise challenges during the study. So that is common to orphan disease studies and cardiovascular studies. Cardiovascular outcome studies. Having multiple vendors, it's very, very similar as well between the two types of studies, but there are many differences as well. In large cardiovascular outcome studies, there is little room for mistakes, for applying protocol amendments or changes after the study starts. Therefore, usually the protocol design needs to be as simple as possible and as close to the standard of care. So due to the large scale of the study and the large number of participating sites, one can imagine it's very challenging applying changes after the start of the study. In orphan studies, we usually see several protocol amendments as a result of feedback from sites or requests actually from regulatory authorities in the participating countries. So there are many more examples, but these are the main ones I can think of right now.
That's a fascinating topic. In some respects, we're orphanizing many of these indications, particularly as Hank has suggested with the biomarker profile that we're attempting to develop. Now, Hank, I know you have a very practical question to ask here.
Yeah, Natalia, going back to the biomarker profiling, given the choice, do most sites and most patients prefer to undergo lumbar puncture or PET scanning procedures?
Well, I would really say that globally the preference would be the PET scan, since it is considered less invasive by most, but it does depend on region and country. As an example, in UK, United Kingdom, both investigators and patients, although they would prefer a PET scan versus a lumbar puncture, if given the choice, lumbar puncture is not a limiting factor to most for study participation. In US, our experience shows that even if investigators and patients may be open to lumbar puncture, caregivers are often the ones opposing to such a procedure and more difficult for them to consent for Alzheimer or dementia patients. In Germany, or in some countries such as Germany, investigators will be extremely reluctant to perform a lumbar puncture assessment to a patient with mild to moderate Alzheimer's disease, as this is considered not part of their clinical practice, and they would usually not agree to do so for the purpose of a clinical trial. And I would say finally, in most Eastern European countries, again, a lampropacture would be equally acceptable as a PET scan if required or dictated by a clinical protocol. But overall, if one would have a choice, they would go with a PET scan versus a lampropacture.
Very impressive expose there, Natalia, which emphasizes something that we all know is true. In international clinical research, it's about knowing culture, language, and standards of care. You can't run the study unless you have a deep appreciation of how folks practice medicine in these different indications. Let me summarize what I think we've heard today, but then we want to ask the audience a poll question. So from Tom, we heard a really excellent illustration of an attempt to link pathophysiology, clinical presentation, and trial development. Tom's appreciation especially emphasizes that the mastery of 3 different levels of sophistication is required regardless of an individual's responsibility. We have to be able to at least be good colleagues to appreciate the science, to seamlessly link the science with clinical care, and to be capable upon drawing upon a wide portfolio of cutting-edge trial designs, particularly for the early phase clinical investigation. Hank added reinforcement to that point of view. As we understand more about the biology of these diseases, we have to appreciate the basic research, link the basic research with the clinical trial designs, basically try to inform ourselves, what are the animal data trying to tell us about the new technology? I think it's a real exciting development for all of us. You cannot be a good colleague in this space. You cannot partner with the folks in drug discovery unless we understand that the technology in place is just, it transcends many of the items we've looked at in the past. At the same time, if you're on the clinical side of the street, we need to have a real basket of different ideas, particularly for demonstrating target engagement, which is so key in some of these earlier studies. Natalia reminded us, as always, that regardless of how deep our appreciation may be for the science and the medicine, someone's got to drive this home. And so it means that it has to be designed, these studies and the technology have to be designed in tandem with really excellent operational acumen. Natalia, you mentioned the matrix team. I think that's the way to go here. Everyone understands that to bring a different talent to the organization, a different focus. You mentioned the importance of the medical monitor. I was struck by that myself in that one program. It also reinforces the need that in order to win the battle in drug development, we have to win 3 things, and we have to win them in sequence. The experimental design has to be correct, there has to be unencumbered execution, and it has to lead to kind of a seamless bioanalysis. So it's therapeutic expertise married to occupational acumen that really becomes the clarion call for moving ahead in neurodegenerative disorders, particularly as we step away into other areas which have all of the uncertainty that Tom originally brought up. Now, ladies and gentlemen, before we end for today, we wanted to bring up a question for you. Obviously, you can tell there's considerable passion and background for us here. We want to know, where do you think we should go on future webinars? So here are our choices. So for future worldwide webinars, what additional topics in neurodegeneration would really pique your interest? Now, this is one of these examples where you can vote early and often, so you can vote for all of these if you wish. Let's go through them. One is, we thought was interesting, do the non-clinical models sufficiently inform the colleagues in clinical research? You know, we do pretty good on primary pharmacology and some of the secondary and ADME-tox issues, but what do you think? Do we have to change the paradigms, or are we doing pretty good? Secondly, as we mentioned earlier, it seems like we're orphanizing most things in clinical research. So what is the impact of orphan indications and the impact on how we conduct clinical trials? Natalia gave us a number of touch points where orphan indications and studies in neurodegeneration would share many of the same attributes. A third one, which of course is one of our good topics, so do current regulatory guidances, do they facilitate or hinder innovative program development. That should be a fun one to have a discussion on. Fourth one on the list is do these unique indications, what kind of issues come up in terms of recruitment and retention, and what might be some of the solutions for managing that? And one that we very much encounter, we do an awful lot of work with companies that are smaller companies and have limited financial background. So how does an innovative clinical and financial management approach, particularly for early phase development, facilitate us asking some interesting questions for novel technology? So we'd like you to vote, please, and we would be very interested to see how you would recommend that we proceed. Go ahead, please. Okay, orphan indications look pretty good, and regulatory guidance is phenomenal. Not a surprise, those are always good topics, but one could see an argument for any one of these, actually, if we just shaped it a bit more. Now, we do have time for questions, and we actually did get one already, which was kind of a good one, about the animal model. So the question is, are animal models informative for trial design? So the way I would say it, I'll take that one, everyone, is so I would say that the modern discovery paradigm that most companies have now is truly remarkable. It's truly remarkable evolution in how we're approaching drug discovery. It is not an amalgam, as we know. It's a composite of a number of different services, just like clinical research. Each of those services have a unique contribution to make. The secondary pharmacology, safety pharmacology, ADME, and toxicology, as we all know, are must-haves for early phase development. And I think I agree with most others that have reviewed the field. We're doing pretty good with that. They are very informative. The primary pharmacology, kind of the MOA story, I believe it's useful because it engages this target. I think that is, first, quite sophisticated. It certainly does help us in the earlier phase investigations, particularly when we have to establish, I call it establishing a handshake. It gets in, it hangs around, it doesn't cause issues in terms of clinical management. It engages target. We know that we have a dose range that's workable. The primary pharmacology can be invaluable. Whether or not they're predictable for ultimate clinical utility, perhaps that's a topic of another webinar. Most in central nervous system disorders would argue that the data suggests that phenotypic screening rather than targeted-based drug discovery has been more productive. But again, that would be a terrific discussion to have with other folks on the line. I see that we're just about near the end of today's session, so Colleen, I'm going to turn it back over to you, please.
Thank you, Dr. Murphy. And we want to thank all of you for joining. We were inspired by the discussion today, and we hope you were too. We did cover a lot of topics, and if you did submit a question that we did not get to, we will reach out to you individually. We encourage you to, contact us if you do have further questions about our conversation. As I did mention at the beginning, we did record this webinar, and we will be sharing the link with all attendees, so keep an eye out for an email from me, most likely tomorrow. For more information about Worldwide, we invite you to visit worldwide.com. We hope you enjoyed today's webinar, and have a great rest of your day.